Hello everyone and welcome to today's session. Today we are diving into the very building blocks of life, which are also called as biological molecules. These are the contents of this chapter. Have you ever thought what actually happens to the food which we eat or why water is so vital for every living cell? In this chapter, we are going to break that down. First of all, we shall identify the chemical elements which build our carbohydrates, fats, and proteins. Then, we shall look at the smaller subunits. Think of these like Lego bricks, which snap together to make these large molecules. We shall also get practical exposure. I will show you the specific test we use to find starch, sugars, and vitamin C in food. We will then wrap up by exploring the amazing structure of DNA, which is the instruction manual of life. And for why water is the ultimate solvent which keeps everything moving inside an organism. So let's get started. First of all, we shall discuss building blocks of life. Now let's look at the skeleton of life. Almost everything in biology is built around carbon. Think of carbon as the ultimate backbone. Carbon atoms love to bond with each other to form long chains or stable rings. This unique ability is what allows nature to build huge complex structures which we call as macromolecules. But what actually goes into these molecules? There are a few essential elements you need to remember. Every biological molecule contains carbon, hydrogen, and oxygen, C, H, and O. However, as we get more complex, we add more ingredients. Proteins, if we talk about proteins, they always add nitrogen and sometimes a bit of sulfur. Nucleic acid, like your DNA, add nitrogen and phosphorus. To understand how these are built, think of a beaded necklace. A single bead is called a monomer. You can see that here, okay, which we call as monomer. So when you string those beads together into a long chain, you create a polymer. As an example for you, remember the CHON rule, okay, this one, CHON rule. Carbohydrates and lipids only use C, H, and O, but if you see nitrogen, then think it is a protein. This is a common exam trick, which is being asked that which molecule contains nitrogen. So if you remember nitrogen, N is for nitrogen and protein, then you can easily get good marks. Now that we have understood what these molecules are made of, let's look at the four main groups of life. I have put for your convenience, for your easy understanding, I have put these into a table to make it easy for you for comparison. First of all, carbohydrates. They building blocks which are the monomers, which are the monomers, the building blocks are monosaccharides like glucose. When you chain them together, you get polysaccharides like starch, cellulose, or glycogen. So polysaccharides like next, I think polysaccharides have been completed. Okay, then we are looking at proteins. Next, we have proteins. These are built from amino acids. When these link, when these link up, they form polypeptides. Think of these as the doers of the body. They make up your muscles, your antibodies, and your enzymes. Then we have lipids, which are your fats and oils. These are vital for long-term energy, which keep you warm. And finally is nucleic acid. The monomers here are nucleotides which build the DNA that carries your entire genetic code. As an exam alert or a takeaway tip for you, do not fall into the non-polymer trap. Examiners love to test this. Strictly speaking, lipids are not true polymers. Okay, lipids are not true polymers. Why? Because they are not made of a long infinite chain of the same repeating unit. Instead, One glycerol molecule joins with three fatty acids to make a triglyceride. And a quick keyword tip is if you see cellulose, think plant cell walls. If you see enzymes, your brain should immediately jump to proteins. Do not get confused about lipids, proteins, nucleic acids, DNA, and all these biological molecules. Okay, we shall discuss that in detail in this lecture. So you should not be confused about it. Okay, you should continue understanding these concepts. This is just a, you can say this is just a introduction of what we are going to cover in today's lecture. Now, the question is, why does the body bother turning small molecules into giant ones? It is because the physical properties change completely when monomers join to form polymers. This allows the body to use the same basic building block for totally different jobs. For example, think about glucose. This is small and highly soluble, meaning it dissolves easily in water. This makes it perfect for transport in your blood or a plant's phloem. It can zip into cells quickly to give you that immediate burst of energy through respiration. But look at cellulose. It is made of thousands of those same glucose units, but they are bonded in straight parallel chains, and this makes the molecule insoluble and incredibly tough. So instead of being fuel, it becomes a building material. Cellulose becomes a building material, providing the tensile strength needed to stop plant cell walls from bursting. Here is a takeaway for your exam. Let's talk about the solubility secret. Sometimes examiners love to ask, why do organisms store energy as starch or glycogen instead of just keeping it as glucose? The answer is osmosis. If you remind the previous chapter, which was about osmosis, diffusion, active transport, you can easily get this answer. You can easily understand the concept behind this answer. If a cell stored 1,000 tiny glucose molecules, what will happen? The concentration would be so high that water would rush in and burst the cell. Because polymers like starch are insoluble in water, they have zero effect on the water potential. They just sit there quietly until they are required, keeping the cell's osmotic balance perfectly safe. So we have looked at the building blocks, but even the best built machine would not run without the right environment. Every single cell in your body needs a specific internal climate to stay functional. First of all, let's talk about water. We call it the universal solvent for a reason. Imagine trying to stir sugar into a dry bowl. It does not react. But if you add water, then it will dissolve. So all your metabolic reactions happen in the water-based cytoplasm. Next, we have salts or ions. These are the cell's traffic controllers. They maintain the osmotic balance so the cell does not shrivel up or explode, and they act as electrical signals. For, for example, your nerves fire by moving sodium and potassium ions in and out. Then there are vitamins. Think of these as the oil in the engine. You only need a tiny amount, but without them, your metabolism would grind to a halt. And of course, oxygen, which is the key that unlocks energy from glucose during aerobic respiration. So, when an examiner asks why water is important, do not just say for drinking. You must mention two things. This is a solvent. Substances must dissolve in it to react. Secondly, this is a transport medium. It carries things in the blood or a plant's vascular system. So always you should also keep this in your back pocket for plant questions. Plants specifically need magnesium for chlorophyll and nitrates for potassium. Magnesium requirement for chlorophyll, we shall discuss that under the chapter head of plant nutrition and photosynthesis. Okay, there we shall discuss that in detail. So you will see that pop up in and fill in the blank question all the time. Now let's zoom in on the quick fuel of the biological world, which are simple sugars. These are small, sweet, and dissolve easily in water. First of all, we have monosaccharides. Think of these as single units. The most important one is glucose. It is the primary fuel for your cells and is usually shaped like a little hexagon. Like a little hexagon. Okay, hexagon. That is the shape. Then there is fructose, which you find in fruit. And interestingly, fructose has the same atoms as glucose, just arranged in a different shape. Now, when you link two of these single sugars together, you get a disaccharide. For example, two glucose molecules join together to make maltose. And if you take a glucose and a fructose and snap them together, you get sucrose. That is the sucrose, which is the white sugar we usually put in our tea. But look closely at the math. When two sugars join, they undergo a condensation reaction. This means a molecule of water is squeezed out to make the bond. That is why the formula for maltose is not exactly 2 times of glucose. So you have to subtract the atoms which made the water. Now let's talk about the giants, which are polysaccharides. These are formed when hundreds or even thousands of glucose units join together. Unlike the simple sugars we just discussed in the previous slide, these large molecules are insoluble in water, which makes them perfect for long-term storage and structure. Look at the three main types of this table, which we summarized in this table. First of all, we have starch. These are long coiled chains of glucose used by plants to store energy. The next one is glycogen. This is highly branched, which allows animals like us to break it down very quickly when we need a burst of energy. We store this in our liver and muscles. Finally, we have cellulose. Instead of being coiled or branched, these are straight parallel chains. They group together to form incredibly tough fibers which make up plant cell walls. This is one of the most common questions in your syllabus. Why store energy as starch? You must remember because starch is insoluble. This is a recap of the previous slide, okay? You must remember because starch is insoluble, it has no effect on the water potential of the cell. So if a plant kept all that energy as glucose, water would rush in by osmosis and the cell would swell or even burst. So starch keeps the cell's osmotic balance safe. And if we talk about the practical exam, then remember the iodine test. Iodine test, we shall discuss it later in this chapter, okay? When we shall discuss about practical exposure, then iodine test will also be discussed. By now, you might be wondering, why does the solubility of a carbohydrate matter so much? It is because the chemistry determines the biological role. If these properties were different, life simply could not function. First of all, let's talk about transport. Because glucose is soluble, it dissolves easily in blood plasma or plant fluid. Think of it as a delivery service. It can be moved rapidly to where it is required the most, like your working muscles or a plant's growing roots. The next one is the storage phase. Imagine trying to fit 1000 loose bouncy balls into a small room. It would be chaotic, okay? If a cell stored 1000 individual glucose molecules, the concentration would be so high that water would rush in by osmosis and burst the cell. So, by packaging them into insoluble starch or glycogen, the cell creates a stable warehouse that stores energy safely. And finally is the structure. Cellulose is insoluble and incredibly tough, as has been discussed earlier, okay? And because it does not dissolve in rainwater or cell sap, it provides a permanent rigid frame. This is what allows a tree to grow hundreds of feet tall without collapsing. So, my dear students, when you are sitting your in your exam, do not just say starch is better for storage. You must use the technical phrase, technical terms. Which one is that? It has no osmotic effect on the cell. This is very technical, and then examiners will really appreciate it. It has no osmotic effect on the cell, or it does not affect the water potential. You have to link the chemistry to the fact that it prevents the cell from bursting. That is the key to getting the higher marks. Now, let's look at the lipids, the fats, and the oils. I want you to pay close attention here because lipids are the rebels of the biological world. Unlike the starch or proteins, we have not studied protein yet. Okay, we shall study it later, but we have studied about starch. We have seen a lipid is not a long chain of repeating units. Instead, it is a very specific assembly, almost like a comb with three teeth. Number one, it consists of one glycerol molecule. It consists of one glycerol molecule, which acts as the vertical backbone, and three fatty acids chains which attach to it. And because of this 3:1 ratio, we call this molecule a triglyceride. We call this uh, we call this as triglyceride because of this 3:1 ratio, three fatty acids, okay, and one glycerol. One thing you already know about lipid is that they are hydrophobic. That is just a fancy way of saying they are water hating. Hydrophobic means water hating. It is exactly why your salad dressing separates and why oil and water never mixes. As a takeaway tip for your exam, do not fall into the not a polymer trap. Okay, I have also discussed it earlier. This is a recap. Do not fall into the not a polymer trap. If an examiner asks if a lipid is a polymer, the answer is a firm no. Lipid is not a polymer because to be a polymer you need many repeating units, but a lipid is just one set structure. It does not grow into an infinite chain. And finally, how do we test for it? We use emulsion test, mixing ethanol and then pouring water. We shall discuss this emulsion test later in this chapter, okay, when we shall find out oil or fatty or oil or fats in our laboratory. So my dear students, next is fats versus oils. Okay, have you ever thought why butter sits solid on your counter while olive oil stays a liquid? It all comes down to the shape of those fatty acid chains we just talked about. First of all, let's look at fats. Okay, these are the properties of fats and these are the properties of oils. First of all, we shall discuss about fats. These are solid at room temperature and usually come from animal sources like butter. Chemically, these are saturated. These are saturated fatty acids. Okay, this means the chains are straight and can pack together very tightly, much like a neat stack of logs. This you can see in this diagram. Okay, this is the chain of a saturated fatty acids, and because they are so tightly packed, they stay solid. Now compare that to oils. These are liquid at room temperature and mostly come from plants, and these are unsaturated. Okay, these are unsaturated, but here is the secret. Okay, their fatty acid chains have kinks or bends in them. You can see that here, kinks or bends. Think of these like crooked branches instead of straight logs. Means these are not straight. Saturated fatty acids have straight chain, while unsaturated fatty acids do not have straight chain. They just can't pack together tightly, and because they stay loose, the substance remains a liquid. And you can also see here that unsaturated fatty acids also have a double bond. Okay, but saturated fatty acids, they do not have, that chain does not have this double bond. As an exam tip for you, if an examiner asks why oils are liquid and fats are solid, the simplest answer is that oils have a lower melting point. So for those of you aiming for top level marks, you need to go a step further. Mention that double bonds in the unsaturated chains create those kinks or those crooks, and these kinks weaken the forces between the molecules, making it much easier for them to slide past each other as a liquid. So we understand what lipids are, but why do cells need them so badly? They're absolutely indispensable for two main reasons. First of all, is cellular architecture. Every single cell is wrapped in a waterproof barrier called the cell membrane. This is made primarily of lipids, specifically phospholipids. If you remember phospholipid bilayer, okay, then you can easily understand this point. So because lipids do not dissolve in water, they act like a high-tech security fence, controlling exactly what enters and leaves your cells. So without lipids, your cells would simply dissolve into their surrounding. Second one is energy storage. Think of lipids as a high-density battery. Gram for gram, lipids contain more than double the energy of carbohydrates. This is why plants store them as oil droplets and why animals store them in adipose tissue or fat cells. Okay, it is the most efficient way to carry a massive amount of energy without becoming too heavy to move. So, in multiple choice exam, you will often be asked to compare energy levels. Just remember, lipids have more energy than proteins, okay, and that is equal to carbohydrates. And if you need extra marks on a theory question, do not forget these 3 points: insulation, protection, and buoyancy. Insulation, it keeps mammals warm. Protection, it acts like bubble wrap or a cushion for vital organs like your kidneys. And buoyancy, it is what helps massive whales float in the ocean. Now we have covered a lot of ground, so let's pause and see how these two essential macromolecules stack up against each other. This summary table, is your ultimate cheat sheet for revision. Uh, as a discussion, both carbohydrates and lipids are built from the same three ingredients. Which one are those? Which are those? Carbon, hydrogen, and oxygen. But look at how they differ in structure. Carbohydrates are built from monosaccharides, while lipids are an assembly of glycerol and fatty acids. You should remember this difference. So when it comes to energy, lipids are the clear heavyweight champions. They have an energy density of about 37 kilojoules per gram. That is more than double of the 17 kilojoules per gram which you get from carbohydrates. This is why we use carbs for immediate fuel and lipids for the long-term savings account. So, students, as a tip for you, do not fall into the common elements trap. If you're looking at a molecule in an exam and you spot nitrogen or magnesium, you can immediately cross carbohydrates and lipids off your list. Okay. If there is nitrogen and magnesium, they are neither carbohydrates nor lipids, because carbohydrates and lipids only use carbon, hydrogen, and oxygen, C, H, and O. And finally, for your practical papers, remember the identity check. If the mystery solution turns blue black with iodine, this is carbohydrate, specifically starch. If it forms that cloudy white emulsion with ethanol, you have found yourself a lipid. If you will master this table, okay, then you can get good marks. And finally, you have covered the half of biological molecules. Once again, I want to make you clarify here that do not be confused with iodine test. We shall discuss this iodine test in detail later in this chapter. Okay. Here today lecture is concluded. If you have any queries, you can freely ask in the comment section. If you have not yet subscribed to this channel, then be a part of this community and hit subscribe button. And do not forget to click on bell icon to stay updated about new video lectures. Till then, I say you all goodbye and best wishes. Thank you.